Prokaryotes are simpler with no organelles and no nucleus. Eukaryotes are complex. They have organelles and nucleus. Alright, endosymbiosis of plant cells. It started out with aerobic bacteria and photosynthetic bacteria, along with ancient anaerobic bacteria. The types of bacteria began forming a symbiotic relationship with the anaerobic prokaryote, and a nucleus began forming. They eventually formed a eukaryote with a chloroplast, nucleic acid, nucleus, and mitochondria. Carbohydrates, you can test Benedict's for monosaccharides and iodine for polysaccharides. Their function is short-term energy. Polysaccharide gives long-term energy. To test lipids, you do a paper bag test, and their functions are their hormones, long-term energy storage, and cell membranes. Proteins, you test with Biuret, and their functions are enzymes and structure components. Nucleic acids, their function is that DNA is a code, and RNA reads codes and makes proteins. Early Earth was much hotter with little to no oxygen, along with an atmosphere composed of various poisonous gases. It was violent, covered in lava, and had frequent lightning storms, and there was no life. Miller-Urey tried to recreate early Earth's atmosphere to see if life could be created by the inorganic compounds of early Earth. They produced amino acids, which are important because they are the building blocks of life. Unfortunately, the simulation wasn't accurate and not all the gases were used. On top of that, no life was actually formed. Dehydration synthesis removes water molecules, builds bonds, forms polymers, and water is the product. However, hydrolysis adds water molecules, breaks bonds, and waters the reactant. They both use water molecules, and both are chemical reactions, and they both require enzymes.